Hey, my name is Noa Oded Elkayam and I'm a team leader in the computational department at Emendo Biotherapeutics. At Emendo, we develop next-generation gene editing tools for genetic disorders. To achieve the optimal gene editing solution for each disease, we use protein engineering to create the best nuclease available for each specific indication. In order to validate each specific editing strategy, the researchers perform many assays ending up with high-throughput sequencing, and we in the computational team help them draw conclusions by analyzing the massive data and outputting relevant statistics and visualizations. To do this, we develop a framework for the researcher to submit analysis requests, which trigger the relevant analysis pipeline and output results to a specific location. Here in this poster, we give an overview of the NGS analysis pipeline. Section B describes the flow from the biological samples on the bench to the final analysis results and conclusions on the cloud. In Section C, we describe the general flow of each analysis, the type of analysis we give support for, and how these analyses are aligned with the editing strategy validation pipeline. In the other 5 sections, D to H, we show output examples for each analysis. Let's start with the NGS pipeline. The researchers prepare DNA libraries ready for sequencing. This can be either manual preps and/or preps generated by our robotic system. These libraries, preps, send to the sequencing, and after sequencing, all data is uploaded to a bucket on Amazon Storage S3. At this moment, each user can upload a request for the relevant analysis he needs to S3 bucket. This upload triggers a Lambda, a serverless service, which quickly performs input validations. Each analysis has its own validation set. Failed validations are sent to the user for corrections, and the moment all validations are passed, the Lambda initiates batch instance, which will run the analysis. At this point, we can have many requests for analysis, and each one contains many samples. Therefore, analyses are parallelized for fast results. When the analysis is done, all outputs are uploaded to a designated location on S3 bucket, and the user is informed. Let's go over the type of analysis we have, which address the validation of editing strategy. First, few words about our editing strategy presented here. In our scenario, we have a disease which, in order to repair, we need to knock out the mutant allele. To do this, we target specific position with SNP, so in that way, when we have a patient that is heterozygote to this SNP, we can cleave only the desired allele which contains a specific SNP identity, ref or alt, which is linked to the mutation. If you look on the left scheme, we show here 3 strategies targeting 3 different SNPs on the desired indication. The best nuclease is engineered for each editing strategy, and we want to validate it works efficiently with no risk for the patients. Note that each step in the pipeline that reports bad efficiency or risk reverts to the engineering step. We start by checking the editing efficiency of our target and continue to validate the SNP discrimination. Then we move to validate we didn't cleave other genomic regions called off-targets by discovery and validation assays. Next, we test our post-treatment effect, analyzing transcripts and expression levels. After passing all strategy validations, we have another assay which fits for each patient its appropriate editing strategy. Now, if you look on the right scheme, you can see the general flow of each assay analysis pipeline. We start by analysis requests containing multiple samples. The first step includes data validation and pre-processing. Then, all sample analyses are parallelized and relevant statistics are reported. At the end, there is a post-processing step which takes all samples and produces summary, comparison, normalizations, and more. Now let's go over the analyses themselves. The first 2 analyses on the top are for gene editing levels of the target site itself. We use CRISPR-2 to get editing patterns for each sample. We have some post-processing that count only the relevant editing we are interested in. We also analyze the SNP discrimination efficiency, making sure we cleaved only the relevant allele. As you can see in the reads themselves, sometimes the SNP is deleted after the repair mechanism, or the SNP identity is not one of the 2 expected nucleotides in the heterozygote cell. It is neither the ref nor the alt. We report the counts of each of these scenarios to give final discrimination score. The two analyses below address the off-targets validation. We first have a pipeline that scans the off-target space and produces a Manhattan plot with the coverage of each potential off-target. These potential off-targets are then examined using RAMSeq method, which check editing levels of multiple applicants at once. This is a massive parallel procedure which sometimes performs around 10,000 editing analyses at once, and there is a post-processing step which summarizes the results and report off-targets that have significant editing levels in the appropriate cells. After verifying editing levels of on and off targets, here you can see the analysis of assays examining post-treatment effect. The top analysis is called abnormal transcript analysis. For this, we use scanty3 to characterize the composition of all transcripts of our indication. The post-analysis compare treatment to control and identify how many new abnormal transcripts are novel and produced due to our treatment. The analysis below on the left shows RNA-seq data analysis using NF-core pipeline. It outputs the coverage of each gene per sample, and our differential expression post-analysis compare treatment to control and compute the p-values and fold change which help filtering genes that were over or underexpressed due to our treatment. The last analysis below on the right present linkage analysis which aims to find what SNPs are heterozygote for each patient and which of the SNPs AlterRef is linked to the mutation. For example, patient 1 is heterozygote to SNP1 and the mutation is on allele 2, so we will treat him with a strategy targeting SNP1 on allele 2. So to finalize, our NGS pipeline helped the researcher get accurate results efficiently on a variety of assays in order to get fast decisions which accelerate their research and development. Thank you.